There are several problems with the way that genomic DNA samples are prepared today. Namely, they can all be summarized into the fact that the way we prepare samples today is not scalable. Each sample requires a significant amount of labor, a significant amount of time, 4 to 6 hours per sample, and even then requires trained technicians with a high level of skill. People have been trying to tackle various portions of these problems over the last decade or two. However, people have been focused on addressing only subsections of the overall sample prep workflow. What there needs to be is a focus of combining all the different steps associated with current sample preparation workflows into a single easy-to-use and automated system. So the world is going to be changed by genomics. It's going to affect so many aspects of our life, from how we eat to how we sleep to better understanding the microbiomes around us, in our hair, on our skin. Genomics is super valuable for just understanding who we are as biological creatures. And none of this is possible without genomic technologies to analyze and inform us about the underlying genomics. And being able to increase the accessibility to those technologies and increase the throughput of that type of science and discoveries is what we're trying to do at Inso Bio. What Inso Bio's microfluidic technology does is it utilizes tiny micro features, sort of like the prongs of a fork, to capture extremely long molecules, such as DNA, in solution. We are able to keep the genomic DNA in solution during the entire extraction and purification process, whereas currently available methods generally have to precipitate the DNA, dehydrate and rehydrate that DNA, which causes loss but also damage to that DNA. What Inso Bio can do is uniquely deliver any size DNA fragments for any type of sequencing platform. Reduce the hands-on time from 1 hour to 5 minutes, speed up the entire process from 4 hours to 40 minutes, and leverage microfluidic innovations to yield higher purity and higher quality sample with less DNA damage. What I believe is that we'll be part of the background. We'll be part of one of those ubiquitous instruments that sits on a lab bench that performs a certain process that everybody sees and recognizes and knows how to use, similar to how a thermocycler is used for PCR. Many laboratories have a thermocycler. We wouldn't be able to imagine going through the PCR process without a thermocycler. So similar to how computers have touched more than just probably 1 billion lives, genomics is likely going to have a similar level of impact. But similar to how computers couldn't have made that level of impact without increased accessibility and further democratization, the same is for genomics. Preparing genomic samples right now is much too difficult even for the best and trained scientists. It's inconsistent and very labor intensive and completely not scalable, and this is what we want to address. So what we see from our system is the capability to not only streamline and automate more of the workflow, but we see a higher quality of DNA being prepared by our system. We're increasing the poly read lengths about 30 to 35% above the average. And this is the primary metric that people use to determine the quality of the DNA during sequencing. Well, right now, if I walk into a hospital and they're trying to analyze me for, say, a particular drug of what to give me, they would analyze me as a male, maybe this height, this age range, 30 to 40 age range, and then they would prescribe me something from that. They haven't taken account who I am as an individual, and I think that's what genetics is able to offer the world. Is each person that comes in for a treatment will be evaluated for who they are from an underlying biological perspective and be given the most effective treatment strategy for their underlying biology.